Okay, so today we are going to continue speaking about the microbiology, the pharma chapter, and also a little bit of biochemistry. So as you can see, what I'm doing, that I'm giving you every day one dose of biochem because I know this subject is not easy. So if I give you small dosages, a little bit at a time, you will get really good immunity and you will be desensitized against it. And it will be more comfortable for you. You will have energy. So for today, the first thing I would like to start with is actually to speak about a difficult subject, which is functional organization of the eukaryotic gene. What? So let's start with the first word, eukaryotic. When we speak about eukaryotic, is it bacteria or us human? Absolutely. Yep, it's us human. So in the human, you remember that there is this, basically our genes are put in a way that they are 5' to 3'. So can somebody tell me what does it mean 5'? What does it mean 3'? Absolutely, absolutely, yes. So if we look at the DNA structure, if we take a look at the DNA structure, we can see the connection between one nucleotide and the another nucleotide is made at a certain bound, which is called the phosphodiester bound. And these bound are super strong, as we said. And they connect the 3' to the 5' end. And by doing that, they make such a stronger contact with it. And when we say, oh, it's a 3' to 5', it means that it started from the 3 carbon on the sugar until the 5 carbon in the sugar. And yesterday I was telling you about like more deeply what that means. But today in a few seconds, I just want to show it to you really quick. So if we take a look at this structure, you're like, what is that? Biochemistry again? Man, have some mercy on me. No. So this is the nitrogenous base. This is the adenine. Basically, when we say a nucleotide is made out of phosphate, nitrogenous base, and sugar, yeah, like ribose in this case. So you can see here the nitrogenous base is bound to the 1' carbon. Here is the 2' carbon. Here is the 3', 4', and 5'. So when we read something in the first day telling us, oh, it's 5' to 3', where is the 5'? Here is the 5'. To the 3', here is the 3'. So this is the 5' end. And if we go all the way here, this is the 3' end. So when we say it's 5' to 3', it means it goes from this point to this point, okay? That's what it means. Okay, keep in mind, this is 2 strands. So between them, there is a connection, which is the hydrogen bound. And between, if we have like 1 strand, between 2 nucleotides on the same strand, we have a phosphodiester bound. What is more stronger, guys? The hydrogen bound or the phosphodiester bound? Phosphodiester, absolutely. Good. So notice here, we have a DNA. This is our DNA. Now I have a question. If I want to check someone's DNA, which tissue I should take from his body? Skin is possible, yes. So let's say I take a piece of his skin, it will work. If I take a piece of his hair, it will also work. Bone marrow will work. Blood. Okay, well, actually, blood, the RBC not really because they don't have the nucleus. But any other neutrophil, for example, or lymphocyte will also have it. Neurons, if I take neurons, can I find the DNA in them, inside of the neurons? The answer is yes, absolutely. So take a look. There is something interesting here. How many chromosomes can I find inside of the skin cell? A skin cell, how many chromosomes will, okay, 46 XY in me. For example, in me, it's XY. So what about a brain cell? A brain cell, how many chromosomes I will find in it? 46 XY. Okay, if you decide to take an epithelial cell from my mouth, how many you will find? 46 XY. Okay, spermatozoa. Absolutely, spermatozoa, how many? 23X. Yeah, 23X or Y? Okay, XY, absolutely. So you notice something. All of our cells have 46 XY. So how the hell a brain cell is a brain cell and the skin cell is a skin cell? Who decides? Well, that's our job today, okay? Today we are going to understand how we get DNA that is like basically the book, and from the book we decide which cell to make and how we make the cells. One of our colleagues today shared an article, by the way, about how about this transplantation of a big heart into a human being. I have to say, I was reading it and was like, wow, this is actually interesting. I highly advise you to read it. It's super cool. This is what we call a breakthrough in medicine, okay? Basically, they took a big heart and put it inside of a human being, and it worked, and the guy is living, and all works perfectly. This is what we do medicine for, guys. It's the breakthrough, the new stuff that happens. Couple of months ago, I saw the renal transplantation from a pig into human beings. One of the most serious problems that always we face is rejection. So why we are not rejecting these hearts and these kidneys from these animals? Why we are not rejecting them? Actually, it's not about the HLA. Well, indirect, yes, it's about the MHC. Absolutely. So the MHC in the pig, it's a little bit similar to a human being, but because we recognize it's not the same, we just canceled it. So when we canceled it, having a heart inside of the body is the same as having an implant inside of the breast. There is no rejection to it. Okay? That's how it works. Okay. So the next thing we have today, okay, we're speaking about the DNA, and we'll speak how we will end up with the protein of need. Okay? No, breast implant do not have any MHC. They MHC free. No MHC in them. Okay? And these kidneys they don't have any MHC in them. So basically the immune system cannot recognize them. And if the immune system cannot recognize them, can he attack them? He cannot, because there is nothing there. Normally when he attacks the tumor cells, he attacks them because the MHC looks suspicious. The MHC one looks like, it's weird. It's not like ours. But if it did not exist at all, can he attack it? The answer is no. Do you remember how cancer cells can protect itself from the immune system? The cancer cell used to protect itself from the immune system by decreasing the MHC. So if cancer can do it, we can do it. Yeah? But we do it for good. Cancer do it for bad. That bitch. Well, anyway, so it's working and it's really, really exciting. I mean, this is some serious stuff, guys. I mean, this is the future. Oh yeah. Imagine we will reach a day that nobody has to wait on the organ list anymore. Or even better, there is no organ list. You want organ? We have organ. You want organ? We have a farm. Problem solved. So let's take a look. We have the 5' to the 3' end. We know what that is. And next we have the cat and the data box. Notice in the cat box, yes, there is one C in the beginning, but otherwise it's full with A and T's. And the Tata box is full with T's and A's. Why we prefer the promoter sequence to be full with A's and T's? Does anybody know? Yep, absolutely. They have two hydrogen bonds between them. I mean the T and the A's, and the A's and the T's. Thus, they are weaker. So, since they are weaker than the G and the C's, the cutting process will be much more easier. Yes, and it will be much easier for binding and initiating transcription from what the DNA, the RNA polymerase. Yeah. So that's how it goes. So let's take a look and speak how this process is happening. Okay, now we want to do transcription. Transcription is converting what to what, guys? DNA into RNA, absolutely. So notice there's few things here I will speak about before I start the whole thing, so I will not waste time later. So first of all, we have this enhancer and silencer region, and we have an enhancer and silencer region. When we say silencer, is the same as saying repressor. We spoke about one repressor yesterday. It was in which topic? The lacabaron. We spoke about, I said that yesterday we spoke about one repressor. The repressor protein was the lacabaron. And even notice, this guy is outside of the lacabaron. And here you can see that the enhancer or the silencer or the repressor is outside the lacabaron. Okay, so look what we have to do with this. First of all, there is something called exons and there is something called introns. Guys, the exons and the introns. Look, let's say I want to make, just for the sake of letting you know, if we have the following sequence: exon 1, exon 2, exon, where is it? Yes, exon 3. Okay, if you have the following sequence, 1, 2, 3, you will get a brain. If you have the following sequence, 1, 3, you will get skin. If you have the following sequence, 2 and 3, you will get, somebody tell me about the cell really quick, because I want any body cell. Come on, some cell. Epithelial cells, nice. Epithelial cells. Okay. You are like, we are not getting it. What do you mean? What I'm telling that we have multiple axons. And depending on which axons you keep, you will get that type of tissue. Okay? For example, the introns, forget about them. The introns are thrown in the garbage. Okay? So we remove this intron and we take these exons together. So exon 2 and exon 3 will be expressed. Let's say you decide not to express exon 1, and we'll speak why later. You will only have the sequence 2 and the 3, so what we will get? Will we get a brain, skin, or epithelium? Epithelium. Okay, let's speak about another case in which, like, introns, of course, we don't speak about them because they are excised anyway, but let's say that you get the following sequence. 1 exons you get, expressing of it, 2 and 3, you express all of them. So what you are going to get? If you express everything, a brain, absolutely. So first question will come to you is, okay, introns, exons. You are telling us that the exons get expressed, exons expressed. It's not difficult to memorize that. But what happens to the introns, and how does our RNA polymerase know that he has to remove them? Well, he does not. It's not the RNA polymerase that remove them, it's actually something else. It's called spliceosome. So there is a sequence, and I will show you the exact question you will get asked on your boards about it, okay? And that sequence goes something like this. It says GTAG. You are like, what? Well, it starts with a GT and ends with an AG. Before I start, I hope all of you know what GTA is. If you don't know what GTA is, stop studying for a little bit, go play some GTA and come back. But GTA is super important for our today topic. Why? Because it's G, T, A, and as always, you end it with a G. So it's GTAG. Okay. So, few points to speak about right now. It will go something like this. If we speak about a gene sequence, and we look up the introns and the exons and all of this stuff, I want to show you how they show you the question. Yes, they might show you a full sequence of gene, which looks something like this. Okay. You are like, oh my God, oh my God, oh my God, what is that? It's difficult, I don't know it, stop. I'm like, just wait a second. Sorry about the neighbor dogs. So they will show you something like this without the upper portion, without the lower portion. You're like, why you are making it so difficult? And they ask you, can you help me please recognize where is the intron start and where is the exon start? Of course, we are humans, forget about the other ones. So if we start looking at this one, okay, let's do it. Okay, so it should start with what? What should the sequence start with? G. But I told you GT, 2 letters, GT. Okay, GT. Now I need to look for an AG. Okay, it starts with a GT and ends with a AG. Okay, so GT. Now I have to look for an AG. A. Okay, there is an AG. So I can say that it stops here. Okay, that's possible to say. Done. So that's one what? That's an intron. Great. Then I can see again GT. So that's the beginning of intron number 2. Then I have to look for what? An AG. A. Oh, here is an AG. So that's intron number 2. So in the exam, they literally bring you a picture of a sequence and they tell you what does this region represent? And you have to say it's an intron because it started with a GT and it ends with an AG. Okay? So I hope it makes sense. Let's just take a look at this one. No, bad sequence. Well, I just hope it makes sense. It's just like they show you the sequence and anytime you see it like starting with a GT, you are like, okay, that's the beginning. Ends with an AG, that's the ending. That's it. Oh yeah, this one. This is even more difficult question. They can tell you, of course they are not that horrible, but they can name lines for you, okay? Like line 1, 2, 3, 4, 5, 6, and like that. And then they can tell you that, actually let me find it first before I tell you to find it, and it will not be existing. GT, okay. GTAG, no, I want a longer sequence. So let me... Okay, let me start from here. Anybody sees a GT? Okay, here is a GT. Okay, here is a GT. So that's a beginning of a what? When I see a GT, it's a beginning of a what? Intron or an exon? Intron. So are intron expressed or not? No, introns are not expressed. They are thrown into the garbage. So we just remove them and throw them in the garbage, okay? So we have GT, oh shit, AG, okay? So that portion is taken and thrown into the garbage. I hope you, actually let me make the writing much smaller so I can like take them and throw them right away. So here is the GT all the way until the AG. This is an intron. Yeah? And then let's keep going. Blah, 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 blah. You keep going. Here's one more GT. Keep going, 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 going. Oh, here is an AG. So that's another intron. Let's take another intron. So, okay, guys, these two are introns. What about the area in the middle? What is it? The area that is not marked is an exon. Is an exon expressed? Yes, it's expressed. So I hope you understood what is an intron and what is an exon. It's all about the sequence. So anytime in the future they show you a picture like this, do not panic. They will tell you the line number. Mostly they will tell you the line number, which is this one. They will tell you, oh, it's at line 2, and you will look at it and they will tell you it's the bulky region. Okay, so it's really obvious, but just be careful of it. If you see it like it starts with a GT and ends with an AG, be like, oh yeah, that one will not be expressed. It will be thrown in the garbage. It's an intro. Okay, so that's how they decide that they wanna throw up an intron. And the ending, it's a polyadenylation signals A, T, A, and it's basically the ending. It's a sign for the ribosomes in the future to tell them it's enough. It's enough. The codon stop here. Don't take more portions. Okay? And the A tail will bind there, and in a moment we will see what is this A tail. Okay, so the promoter is here. The RNA polymerase bind here. He does his job. And next we have the exons and the introns, and that's still a pre-mRNA after the transcription process has already occurred. Obviously, we don't want it. We do not want the mRNA, we do not want the intron to be there. So we have to take them out. How can we take them out? By a process called splicing, okay? So in the near future, I will be telling you about the spliceosome and how he does that. But for now, just trust me that they are going away, and you will have one sequence for one protein that will be what? Translated. So we will have a mature mRNA that we will be translated and will get a protein out of it. Notice, please, that the mature mRNA has so much A's in his butt and a 5' cap. So the 5' cap help him to recognize the what? Actually, it does not help him. It helped the ribosome to recognize him, okay? So the 5' cap help the ribosomes to recognize the mature mRNA. While the poly A tail, it's really important because it protects the mRNA from damage. Let me tell you how. Well, okay, let's say that this is, let's say that this is our mRNA, and now we are going to make a tail for it. The tail will be blue. It's just like adenine, 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 adenine. It's just like for just a short period of time, and then it will be thrown into the garbage, okay? So it looks like this. The tail is super long compared to the mRNA. So, and of course we said that it has a cap, and the cap was important for the recognition by the ribosomes. So, guys, if there was an endonuclease here, let me make an endonuclease in the shape of a Buckman. If there is an endonuclease here, will it eat the tail that is really, really long, or it will go and eat the mature RNA that is here? Of course it will eat the tail. It has more chance of eating the tail. Why? Because the amount of tail is much more than the amount of mRNA. So it's easier for him to eat the tail. Yes? Okay. So the next thing we have is like about the mRNA, and there's like few points I want to speak about before I forget about them. The 5' cap is super important when we speak about the what? When we speak about the recognition by the ribosomes, as we just said. Okay, so the next thing you need to know about is the regulation of gene expression. So can someone tell me an example of a repressors that we spoke about already? A repressor. Okay, yes. Done. Absolutely correct. So that one was the lac aberon, yes. And now we are going to speak about some regulation of gene expression. We have, first of all, a promoter. What is the promoter? This region, the tata box and the cat box, yeah? And it's the site where the RNA polymerase 2 and multiple other transcription factor bind to the DNA upstream from the gene locus, which is this gene locus. It's upstream from it, like away from it, and it's full with adenine and thymine, as we said, with the tata box and the cat box, which differ between eukaryote and prokaryote. And we said the most important thing for that region was to start and initiate the process of the transcription. As we said, it's an RNA polymerase, so it's a transcription. If I was saying about the DNA polymerase, it will be translation. So you must be comfortable with the difference between these two. So the next point we have to be comfortable with is a promoter mutation commonly results in a dramatic decrease in the gene transcription. Why? Because imagine this. If our promoter is not working, this area is ineffective. Can we transcribe anything in the whole gene? The answer is absolutely not. Our whole gene will be messed up. It's just like impossible. It will be completely useless. Yes, absolutely. Good. Somebody is asking, can we have tata? Can we have only tata gene or only cat gene? We actually, are you asking if we have only cat or tata? Well, here is the thing. It's actually all tata, which means it's like TA. But yes, it's possible to have both. Notice here in the cat box, there is only one C here. Yes? All of the other sequence in both of them is A's and T, and that's the point here. The point that this sequence is full with adenine and thymine that have two bounds. You're like, okay, what about this 1C? Well, this 1C is the most initial portion, so it does not really matter. But the other portions, yes, they do matter because the DNA polymerase has to bind to something, okay? So yeah, we can have both. An enhancer now. An enhancer, it's a DNA locus where the regulatory proteins bind and they increase the expression of the gene on the same chromosomes. So the enhancers will lead to more expression of the gene. When I say more expression of the gene, gene expression is converting the DNA into what? What is that? Can somebody tell me? Yep, absolutely correct. Yes. So the next thing is when we speak about a silencer, it's a DNA locus where the regulatory protein, the repressor bind, which decreases the expression of everything. Okay? So an enhancer and a silencer, these guys. And they say an enhancer and silencer may be located close to, far from, or even within the introns, the genes whose expression they regulate. For example, if we speak about the lac aberon, it was located outside of it. It came from the outside, but it worked inside of the lac aberon, inside of the gene. So now let's speak about how do we back and prepare an RNA to make a protein out of it. So okay, the silencer is The silencer. I did not get it. Silencer AATAA. Are you asking that is a silencer the same as data box? The answer is no, it's not. But is the silencer working at the data box? The answer is also not always. Basically, it can work in any region. But if you are asking, is the sequence of an enhancer full with T's and A's? If that is the question, the answer is also not. So the RNA polymerase work at the area that is full with A's and T's. But here you can find many G's, many C's. It does not really matter. Yeah, and the area after it the same. Okay. So let's speak about the processing of an RNA inside of eukaryotes. So to start with, we have this mRNA. We need to protect it, and we need to put a hat for it. Why do we put the hat or the cap? Why do we put the cap, guys? Why I told you we put the cap? There was a reason we put the cap on. Well, basically, we put the cap on for... No, the cap was not for... it's for recognition of ribosomes. The protection was the tail. Yes? Protection was the tail. The hat was the recognition. So initially, transcription is called heterogeneous nuclear RNA. So this HN RNA is then modified to become an mRNA. The mRNA is the best of the best. It's what we have in the end to make a protein out of it. Okay? So the following process occurs in the nucleus, which is what? If you get a question telling you, where does post-transcription, where does transcription occur? First of all, where does transcription occur? Somebody tell me. Transcription. Transcription is the process of converting DNA into RNA. Where does it occur? It occurs at the nucleus, obviously. Okay, so if they ask you where does post-transcriptional modification occur, please write also nucleus. The following process occurs in the nucleus, which is the capping of the 5' end, which is addition of this thing. It's the 7-methylguanosine cap. So this cap is important for the recognition by the ribosomes. Also, we add a polyadenylation, which has a lot of A's, to the tail, which we call the poly-A tail. More than 200 adenines are put in there. And why the tail is important? For a protection. And after that, the spliceosome comes and remove all of the introns from the place. So we cap it, we tail it, we splice it, and it's ready to go. Okay? So we have an mRNA, we put a hat on it, we make it a tail for it, we cut the introns out of it, and it's ready to go. And all of this process, where did it occur? Where did this process occur, guys? Come on. Absolutely, yes, it occurred in the nucleus. So, mRNA is transported out of the nucleus to be translated in the cytosol. Okay, so we take the mRNA from the nucleus to be translated into the cytosol. Which organelle is responsible for the translation? The ribosome, yes. So the mRNA equality control occurs at cytoplasmatic processing bodies, the B bodies, which contains exonucleases, decapping enzymes, and microRNAs, mRNAs can be degraded, blah, 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 blah. Let me explain what happens. So, inside of the nucleus, what did we do? Inside of the nucleus, we did the transcription, the post-transcriptional modification, which was what? Putting a cap and making a tail, yes? And also, one more thing, what did I forget, guys? Cap, tail, what else? What should I do for him? To make him ready for his bride, Yes, I should remove all of the introns out. Yes, should remove and exclude all of the introns out of it by splicing. Yes, I splice out all of these guys. Okay, done. So let's say he's perfect now and he's ready to go to the cytosol. It will go to some place. It's a processing center. Basically, you know, this big processing center for Amazon. They make sure that you did everything correctly. Let's say that I did not do it correctly. I made a mistake. Should I keep this mRNA or kill it? Yep. So the B body is really nice when the stuff look how he wants. Okay? If it's normal, it will just go through no problemo. It's just a processing center. But if one of these mRNA looks fishy, be like, oh, I see. So you think you are smart. You are having different sequence that will be leading to a dysfunctional protein. Not on my shift. It will do what? It will use its own nuclease, which will destroy the nucleotide. It will decap it, remove the hat, and basically kill the shit out of it. Okay? The mRNAs may be degraded or stored in the B bodies for future translation. So if they are good, they are like, okay, the good guys goes here, and the bad guys goes there. Okay? So I'm bad at this, but I'm trying to make a fire. Okay? So here goes the good guys, and the bad guys get killed at the processing center of the cell, which is called what? Exactly, it's a policeman. More like a psychopath, but okay, yeah, a policeman. Policeman will do that. Yeah, decap people and splice them. Yep. So, polymerase does not require a template. What is this polymerase? Well, the making of the tail, and we said that the tail is made of a lot of adenines. To make it, we need a polymerase, yeah? But this polymerase does not require a template because, come on, how difficult is it to make plenty of A's next to each other? You're like, okay, it does not require a tail, but how does it know that it should make the tail, yeah? How does it know that the tail should start here? Because it sees this sequence, the AAUAAA, which is seen here. So when it sees this sequence, it started adding the polymerase, starting adding plenty of A's to the tail, which help in the protection of the mRNA. Okay? So that was all about the functional organization of the eukaryotic gene and how the regulation of the gene expression occur and how the RNA processing occurs. So now I will be speaking next about the RNA polymerases, and hopefully, according to my plan today, we should speak also about the splicing, and we should speak about introns and exons. Then we'll speak a little bit about the topic of pharma microbiology, and tomorrow we'll be finishing all of the molecular biochemistry, which you don't feel it, but we only have 2 pages left of it. And we'll be done with molecular biology. We'll jump into cellular, which is much more easier. So that's how biochemistry is going to be with me, one small dose every day. So, speaking about the RNA polymerase, eukaryote, prokaryote. Eukaryote, it's us. Prokaryote is bacteria. So the eukaryote, us, who's more complicated, us or the bacteria? Yep, we are really complicated. Yes. So we have 3 RNA polymerase. The bacteria has one that does everything, okay? But in us, we have 3. Why do we have 3 of them? Well, I don't know. Probably because we have to memorize all of them. But the 3 goes something like this. We have RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. The first one, RNA polymerase 1, make rRNA. Before I speak about that, I want you to know one thing. Where does all of the proteins in our body comes from? Which organelle makes all of the proteins inside of our body? No, the nucleus is not it's not an organelle. It's not an organelle. The ribosomes make all of the proteins inside of our body because they do which process? Which process is done by the nucleus? The translation, so it makes us plenty of proteins. Okay, so I agree. So what makes the ribosome himself? Well, ribosome himself is made out of a protein. So since he is made out of a protein, who makes him? Who comes first, the chicken or the egg? Come on. Interesting question, yeah? It's not philosophical, it's actually medicine. So there is a little organelle inside of the nucleus called the nucleolus, okay? Nucleolus. Yes, so it's the nucleolus. It's a little molecule inside of the nucleus. So let's take a look. You can see this is the this is the yes. So this is the nucleus, and there is a little nucleolus inside of it. Does anybody here speak Latin? If you do, raise your hand and tell us what does a nucleolus mean. If nobody, I will tell you. Well, nucleolus means the room of the king, okay? It's the room of the king. Well, what do you mean by the room of the king? Well, basically, if you have a big castle, inside of the castle, you have the main room, yeah? Which is the room that is for the king and the queen, yes? Back in time, the old Romans who used to speak Latin used to say, oh, let's go to the nucleolus, which is the room of the king, okay? Anyway, so it's just some heterochromatin that exists inside of the nucleus. So inside of the nucleolus, we have something called rRNA, which makes the structure of the ribosomes. And it's made by the help of RNA polymerase, which is located in the nucleolus. Cool story. So we have a cell. Inside of the cell, we have a nucleus. Inside of the nucleus, we have a nucleolus. A nucleolus make what? rRNA. Yeah? And the rRNA makes what, guys? Come on. What does the rRNA make? Ribosomes. Absolutely. It makes the ribosomes. Yes. So if someone asks you in the future, where are the ribosomes made inside our body, what you will tell him? The nucleolus, absolutely, good. And RNA polymerase 2 makes mRNA, which is the one we keep hearing about, yes? So, yes, it's the main one. I wanted to speak about 3, but leave it until we reach 3. So let's speak about mRNA, the massive one, and the micro one. The micro one will keep it for its time will come. And something called the small nuclear RNA. These minor ones will have their time in the near future, but for now, let's focus on our mRNA. So that mRNA was the one that used to go to the ribosomes and get it translated to make us proteins. Finally, the RNA polymerase 3 makes us the most tiny portion of the rRNA. And anytime, guys, you see rRNA, what does the little r stands for? Absolutely. So far, we have 2 RNA polymerases responsible for making us the structure of the ribosome. They were 1 and 2, okay? They were, no, sorry. They were, somebody wrote 1 and 2 and I become biased. It's 1 and 3, 1 and 3, okay? So 1 and 3, because 1 made rRNA from the nucleus. And the other one is the 3. It's made the what? The 5S rRNA. Okay. And also it made the tRNA, which was responsible in transporting the amino acids around. Okay. No proofreading function, but can initiate chains. RNA polymerase 2 open DNA, add the promoter sign. promoter site. What is the promoter site? Does anybody remember what was the promoter site that the RNA polymerase 2 works at? Yep, the cat or the Tata box. Absolutely correct. 1, 2, 3 are numbered in the same order that their product are used in a protein synthesis. So the rRNA is to make the ribosomes, the mRNA goes to the ribosomes to be translated, and the tRNA brings the amino acids to the ribosomes to be added into the sequence. So now this is important. Death cap mushroom. Guys, you need to be really careful when you eat wild mushroom, okay? If you get a patient in USMLE that he ate a wild mushroom that his friend advised him to eat with fava beans, it's actually not about the fava beans, but this thing, it's called the what? Black death mushroom, okay? Well, or the death cap mushroom. If it's called death cap mushroom, it has death in the name, you should not eat it. So this guys is super toxic. When we say toxic, it can kill you. But the question comes, how it can kill you? Well, first of all, let me teach you a trick, guys. How to know that you have a death cap mushroom? Oh my God, I'm becoming not a doctor, but something else. Can you see what it has here? It has like something on its neck. Yeah, it has like something on its neck. When you see this wrap around its neck, it's highly specific for the death cap mushroom. Notice, it's also having it again on its neck, And this one. See, it has this on its neck. It's highly specific for the death cap mushroom. It always have it. But, you know, I prefer buying my mushroom from the supermarket. I'm scared of this stuff. So, let's speak about it, how it kills us. Well, it kills us with the same way some antibiotic work. So alpha amantine is a toxin that is found in Amantia phalloides, which is the death cap mushroom. It inhibits our RNA polymerase 2. And does anybody remember where did the RNA polymerase 2 bind on in the gene? Absolutely correct. It was the Tata box. It was the Tata box or the cat box. Absolutely. Okay. So when they inhibit that, there will be no transcription anymore. Yes? If you have absence of the transcription, can you after that do translation? No. So can you make proteins? No. So what will happen to the cells that get the toxin? Yep, all of them will die. Absolutely correct. So guys, I'm a stupido walking in the woods and I see some mushroom and take it from the ground and eat it. Which organ is going to be affected first? Yummy. Absolutely correct. Logically, it will be the stomach and the intestine. Yes. So if it's the stomach and the intestine, I'm going to have dysentery. Well, what is this dysentery, guys? What does it mean, dysentery? Yes, absolutely. It means a bloody diarrhea. So I'm going to get a bloody stool, and then what happens to the food after it goes inside of your small intestine? What will happen to it? It will start becoming absorbed. Yeah, it will start to become absorbed. And when we absorb something to the blood from the intestine, where do we absorb it to? To the liver, absolutely, because remember, all of the GI organs go through the portal vein to the liver. So the next organ to be affected will be the liver. So the liver is going to get intoxicated, so you get severe hepatotoxicity if ingested, and it dies. Okay, so this substance is highly toxic. Should we consume it? Logically, no, but we as a doctor saw that it's a really good killer, so we started to give it for the patient. Why the hell would we give the patient such medication that inhibits the RNA polymerase and kill the cells? Cancers. Absolutely correct, cancers. So there is one chemotherapeutic agent called actinomycin, also the actinomycin, which inhibits the RNA polymerase in both prokaryotes like bacteria and eukaryote. Because it works on us, it can be used for the treatment of some forms of cancers. That's why chemotherapy sucks, because it's hepatotoxic, it can lead to dysentery, and it can have systemic effects. The next one is in the prokaryotes, it's much more easygoing. Why? Because we only have one RNA polymerase. Okay? It has multiple subunits which do basically everything, so you don't have to know what they do. But the RNA polymerase converts the RNA into DNA, correct? No, absolutely, good. So they convert the RNA polymerase converts the DNA into RNA. Okay, nice. So now we have the RNA, it goes to the ribosomes and it will get transcribed, make your protein, cell living happily ever after. In the prokaryote, this RNA polymerase can be inhibited by some drugs, like rifampin, which is used in Mycobacterium tuberculosis, or rifabutin, which is also used for TB. So they inhibit the DNA-dependent RNA polymerase in the prokaryote, which lead to the death of the prokaryote. So we use it as an antibiotic to treat bacterial infections. Okay, nice. I hope you understood that. That was an interesting topic. I spoke about a lot of things, I agree. But now we'll have a little break for 10 minutes. We'll come back and we will continue. So see you guys in 10 minutes. Yes, we do. Hello. Hello everybody, welcome back. So let's continue with our lesson. So the next thing we will be speaking about will be regarding the splicing. So let's speak a little bit about splicing. The idea about splicing was to remove what? What we had to remove during splicing? The introns, absolutely. We remove the introns. And when we remove the introns, we leave out the exons. And how could a spliceosome recognize the beginning and the ending of intron? How he can say, oh yeah, that thing is an intron, so I should take him out. The GTAG. Well, you are totally correct. It's GTAG, but that's if we are speaking about DNA. But to be totally fair with you, the process of splicing is supposed to transcriptional modification. And the transcription is the process of converting DNA into RNA. So if in DNA it was GTAG, in an RNA it will become what? It will become G. Can we have a T in a RNA sequence? Can we have a T? Absolutely. GUAG. Okay? So you notice here the spliceosome is working from the GU until the AG. Yes? And that helps, he spliced that portion out. So let's see what they say about this. So, splicing of a pre-mRNA, a part of the process by which the precursor pre-mRNA is transformed into a mature mRNA. So we need to maturize the mRNA. Remember, to maturize it, we had to do three things. We had to cap it, put a cap on it, and after we put the cap, we had to do something else. What was it? Does anybody remember what was the next thing after putting the cap? Yes. So after putting the cap, we had to like install the tail on, and other than the tail, we also had to do what? Splice tail hat. That's it. Okay. So alteration is in the SNRMB or the SNRP. You are like, what? What? SNRP. You're like, but it's not helping. What is that? Well, it's basically the sequence that help us to splice, the spliceosome. So any alteration of this SNRP, which is the spliceosome basically, can lead to clinical diseases because of poor intron excision, yeah, poor splicing of the introns. For example, it can lead to spinal muscular atrophy, okay? You're like, wait a second, what is spinal muscular atrophy? Well, do you remember a disease due to Clostridium botulinism? When we speak about the Clostridium botulinism, I told you that if the baby eats the honey that has the spores, he can have what? Which disease he can have? The flaccid paralysis. Absolutely, he could have the flaccid paralysis. So that's what I'm speaking about right now. So this baby can have the flaccid paralysis. I totally agree. But also there is few more points you need to keep in mind. This process, part of a process, other disease that is not infectious that can lead to this include the following. If we speak about the spinal cord, let's make a spinal cord here. And here we have this posterior column. We have this thing that looks like a butterfly. And we have here the anterior horn. So you don't have to know this right now. And I don't expect you to know this right now. But in the anterior horn, there will be the neurons that are responsible for the motor function, okay? And in case of SNRP problemo, there will be decrease in the spinal muscular neuron protein development, which can lead to degeneration of the anterior horn of the spinal cord. If the anterior horn are gone, our motor neurons will be gone. If we have no motor neurons, we cannot move. We cannot move, we get hypotonia, and that will lead to floppy baby syndrome, but it will not be due to an infectious disease. In contrast, it will be completely congenital, okay? Yep. So that is the one. So the next one goes something like this. We have something regarding, anti-U1 SNRP antibody is associated with lupus, mixed connective tissue disease, and other rheumatic disease. It's a marker, guys. The anti-U1 SNRP antibodies is used as a marker, and it's really famous for so many diseases that they use it as a marker in. So keep it in mind. Okay. Since we are done with this one, we can move forward and speak about the next note. So the next note says something like this. We have something that says, well, according to all of the problems we spoke about, we have to learn how we can do the splicing. So the first area was to recognize the GU, the AG, cut them, and splice that portion out and get it outside. And you can see that who is helping us in this with this task? The SNRPs. So the SNRPs were there to help us get it out. So they write, primary transcript combines with this SNRNB, which is the SNRP, and other proteins form the spliceosome. So spliceosome is many particles, including the SNRPs, which help take that portion out. And after that, they make this Larray shape or loop-shaped particle that will be removed and thrown outside. So the introns goes to garbage. And we are left out with 2 exons, which goes to the mature mRNA. We put a cap on them, we put a tail on them, and they are ready to go to which organelle? Which organelle are they ready to go to? Yep, the ribosomes. That's it. So that is the whole process of splicing. Let's speak about introns versus exons. Exons contains the actual genetic information coding for the proteins. And we said exons gets expressed, introns gets thrown away. The introns do not code for the proteins, but are important in the regulation of gene expression. Okay, because they are like, yes, take that piece, no, take that piece. Remember when I told you about 1, 2, and 3? And for example, 1, 2, 3, if they are together, they code for the brain. If 2 and 3 are together, they code for the epithelium. So stuff like that. So it's the introns who decide which portion to stay. Different exons are frequently combined by an alternative splicing to produce larger number of unique proteins. So it's depending on which form of splicing occur, tell us which type of a protein we will get. For example, you notice here, we have the exact same DNA encoding for a protein 1, 2, and 3. And all of that depended on which exons got expressed. So here it was 1, 2, 3, 4, 5, 6. Here it was 1, 3, 5, 6. Here it was 1, 2, 3, 4, 5, 6. Okay? So according to which arrangement they are done, we will have a different protein. Alternative splicing can produce a variety of protein products from a single heterogeneous nuclear RNA sequence. For example, it can make us the transmembrane versus the secretory IgAs, or it can make us the thromboyosin variant in the muscles. We have different types of strobomycin inside the 4 muscles. Dopamine receptors in the brain. We have the D1, D2. Host defense evasion by tumor cells. Okay. So basically, to be totally fair with you, it all depends on which sequence did the splicing occur. If we took that portion, we get that disease. If you get that portion, you get this. If you did that sequence, you get that protein. If you did that sequence, you get that protein. It all depends on which splicing you did. And you must know that during the splicing problem, we can get this spinal muscular atrophy. And it's super serious disease with degeneration of the spinal cord. And we are going to speak about it again. And they would like to confuse you with this disease. With which other disease, guys? Which other disease has similar symptoms? I mean, floppy baby, except botulinism. Which other infectious disease have similar symptoms? Infectious, something infectious. Well, I hope you are thinking about poliovirus. It's polio. Polio also destroyed the anterior motor neurons of the spinal cord, which also leads to lower motor neuron symptoms, which lead to exact same symptoms as spinal muscular atrophy. So this is the spinal muscular atrophy. As you can see, it's a defect in the nerve assembly. Okay, done. Nice. Okay, so now we can go back. Since I finished the chapter from biochem, what I wanted you to know for today, which was the following. So let's take a quick look at it. So I wanted you to learn this page, and this page. Okay, 3 pages. That is done for today. For tomorrow, we finish the last 2 pages from molecular biology, and that will be it. But for today now, I will want to continue with you a little bit with microbiology.pharm. So yesterday, we were speaking about so many drugs working at the protein synthesis. And we were started to speak about the aminoglycoside. We finished it, but we did not start with the other drugs. I hope you watched the videos, and now we are going to speak about tetracyclines. So, starting with tetracyclines, can somebody tell me what is a contraindication for tetracycline? What is a complete tetraindication? Yes. Pregnancy or being really, really young, yes? Basically a baby. A baby should not take tetracycline, not a good idea. Okay. So when we speak about such patient, the patient with such diseases tend to have some serious problem. For example, from the tetracycline, they get poor bone growth. Okay, poor bone growth. So tetracycline, we have tetracycline as a subtype, doxycycline, and minocycline, and dimeclocycline. Dimeclocycline you never heard about because we'll learn it in endocrinology, not in antibiotics, because it has some side effect. So mechanism, they are bacteriostatic drugs, which means they do not actually kill the cells, they just halt its growth. And they bind to the 30S subunits and prevent the attachment of the aminoacyl tRNA. So they prevent the attachment of the aminoacyl tRNA, which is a molecule, ridiculously enough, I will be telling you about tomorrow, which help the attachment of the tRNA sequence with is a protein to bind to the mRNA to bring us what? To bring us the amino acid. So if this process is inhibited and it's not happening, can we do translation anymore? Is it possible to do translation anymore? Absolutely not. We cannot do translation. That's the problem. So if we cannot do the translation, we will not have proteins. If the bacteria does not have proteins, it will die. Okay? So it binds to the 30S subunits and it prevents the attachment of the amino acid tRNA. It has a limited CNS penetration, thus we don't regularly use it as an anti-meningitis drug. It's not regularly used for that sake. And for the next thing, we have to speak about the fact that this drug, doxycycline, is fecally eliminated. That's really good. Why? Because most antibiotic, how they are eliminated, guys? How most antibiotics are eliminated? Absolutely, yes. So most drug overall are eliminated fecally. So it's a really bad idea actually to Yep. So most of them are eliminated fecally. Thus, it's kind of a really bad idea. Sorry, I'm really sorry. Most of them are eliminated renally. I'm thinking about it. No, it's actually renal. Yeah, they are going out from the kidney. So if the patient has kidney disorder, it's a bad idea to give him any of this medication like aminoglycoside. So you would like to give him tetracycline. Do not take tetracycline with milk. And because it has calcium, or antacid because they also have calcium and magnesium, or an iron containing preparation because of what? Well, basically all of these drugs, I mean tetracycline, calcium, magnesium, are divalent cations, which means they have positive charge. And it's not any positive charge, it's the +2 positive charge, which means they cannot reabsorb from the intestine together. Yes? So if you drink it with milk, you will have less reabsorption of tetracycline, which can make the drug completely useless. Okay? So to make it simple, do not take it with tetracycline and do not take it with iron containing substances. So clinical use: Borrelia burgdorferi, which was the cause of Lyme disease, Mycoplasma pneumonia as well. A drug ability to accumulate intracellularly make them very effective against the rickettsial and the chlamydial infection. Remember, they were the type 1 drug, doxycycline. We kept saying doxycycline. Actually, we used to use it a lot in treating acne, but we don't like to use it anymore. Does anybody know why? Well, the answer is that because it can lead to some side effects, such as the teeth thing, you know? It can lead like to discoloration of the teeth, make them yellow. So A drug ability to accumulate intracellularly, we said it works in intracellular organism used to treat acne. Doxycycline is effective against community-acquired MRSA, not hospital-acquired MRSA. Be really careful about this. Let me tell it again. It's working against community-acquired MRSA, not hospital-acquired MRSA. If you had to choose between hospital-acquired MRSA and community-acquired MRSA, which one is the worst? Which one is the worst? The hospital-acquired, obviously. And adverse effect, GI distress because of killing of our bacteria, discoloration of the teeth, and inhibit the bone growth in children. Can have photosensitivity. That's why, if you take a look at doxycycline, if you take a look at the doxycycline, it mostly comes in tablet and they tell you, keep it in a dark room. So, because it's photosensitive also, the drug himself, I mean. In addition to the fact that he is a drug sensitive, it makes you photosensitive as the patient. Be really careful about consuming bad tetracyclines. I mean, when I say bad tetracyclines, I mean like outdated, okay? Why? Because the outdated tetracyclines can lead to Fanconi syndrome. Make a note about that. So Fanconi syndrome can be caused by what? By, how do you say that in English? Expired tetracycline. Expired tetracycline. So expired tetracycline can lead to Fanconi syndrome. Make a note about this. They do ask about it. Okay. Mechanism of resistance. Basically, they are like, oh, the bacteria used to uptake this tetracycline. Okay, so I will stop. They stop taking it in, and they make an efflux pump that kicks out the drugs. You are like, seriously? Are you playing with me? And does anybody remember which organelle or which particle inside of the bacteria is responsible for the resistance against our antibiotic? Which portion inside of the bacteria is responsible for the resistance? The plasmid, absolutely, the plasmid, yes. Okay, so they are plasmid encoded transporter pumps. So guys, before I move on, I want to tell you about some exciting news. Well, some people might think that is really bad, and it's kind of bad, but still, we look at it as a doctor, not as a sensitive people. So let's take a look at this. The COVID situation in the US is really bad. You can, it's around the world really bad. I mean, it's the first time we have numbers in the millions. But speaking about the new variant, the Omicron, guys, does Omicron have high virulence, which means does it kill more? Is it more harmful or less harmful than the previous variant? It's much less harmful. Does it and has much less severe symptoms? Okay, what about the transmissibility of it? Super high. Guys, I was reading an article yesterday. This thing is ridiculous. He's super fast that it's kind of like measles, which is like the fastest thing we have it spreading. Well, that means that we, in the following couple of weeks, we need to expect millions and millions and millions of new cases. Is that a good thing or a bad thing? I see not, most of people are not answering because, yeah, that is the answer. It depends. If we speak about the fact it will help us to get rid of COVID, yes or no? Absolutely. It will help us to have a herd immunity much more faster. But we are still afraid of one thing, the hospitalization stuff, which means like a lot of people will need hospital. Yes, we have 2 million cases per day. I mean, come on, is that just a ridiculous number? Even for the US, which is like have really good medical system, to have almost 1 million cases per day, this is a huge number. If you say even like 1% hospitalized, this is a huge number. We are speaking about 9,000 people hospitalized each day, which is just a lot. But still, if the hospitals will be able to continue with this, the prediction is telling us that by June this year, the numbers of the infected people will be super low, okay? Because we will be actually, for the first time, established herd immunity. Everybody will get it. So that's why you notice, maybe some of you who live in the US notice that the CDC is not caring anymore. I mean, back in the time, they used to say like, put masks, do this, do that, no. And now they are like, okay, well, yeah, you should put mask. Yeah, wink, wink. Because they want to achieve herd immunity and get rid of it. If you are vaccinated, don't worry about it. Even if you get it, the symptoms are super mild. Exactly. They want it to spread it, basically. The symptoms are super mild. The patients are not going to have it bad, and that's good. According to what I read, like most people have just like a rhinorrhea from the nose, a little bit of cough. That's it. No actual dyspnea, which is amazing. The dyspnea sucks. No dyspnea. Okay, good. Hopefully this will be beginning of the end. The next one, the tigacycline. Tigacycline, it is one of the tetracyclines family, but it's a little bit different because we use it for specialized type of organisms that are mostly resistant. Remember, a tetracycline, it's one of the A30. So absolutely. Tigacyclines. Sorry, not tigra. Tiga. I just say tigra because I'm used to the tiger's theme in Sketchy. So yeah, it's a tetracycline derivative that binds to the 30S, which inhibits the protein synthesis. It's also bacteriostatic. It's a broad, it can be used against what? Everything. So it can be used against the broad spectrum anaerobe, gram-positive, gram-negative coverage, multi-drug resistant organism like MRSA and this horrible guy. Yes, absolutely correct. The vancomycin resistance enterococcus, or infection requiring deep tissue penetration. Okay, so adverse effect, nausea and vomiting, nothing serious, but this drug is not that old actually. So we should use it more often, yeah? Should we use it more often? Give it to all the patients since it covers literally everything. It's an amazing drug. We should, yeah, give it to everybody. No, we should not. We should keep it for the most severe patient we have. We don't want to have more resistance. Okay, chloramphenicol. They block the peptidyl transferase at the 50S ribosomal subunit. It's a bacteriostatic. Remember, I told you, baby T-deal. Yes, it was the baby T-deal. Here it is. So it used to work on the baby T-deal transferase. It's a baby. Why baby? Because it leads to gray baby syndrome. Okay? It's not much. Yeah, I told you, it's quite not an old drug at all. It's a new drug. I'm not sure which year, but I know for sure that it's new. But no, it's not that new, but it's new. Chloramphenicol, it blocks the peptidyl transferase at the 50S subunits of the ribosomes. The clinical use include meningitis, Haemophilus influenza, Neisseria meningitis, Streptococcus pneumonia. All of these organisms have one thing in common. They all are encapsulated. Also, it can work in intracellular organisms, like, for example, rickettsia, which lead to Rocky Mountain spotted fever. It has a limited use because of the toxicity. It's mostly used in the developing countries because of low cost, especially for meningitis. Write a note about this. In the developing world, it's still used for meningitis. In the US, it's not used for meningitis. Write a huge note. The only disease we use chloramphenicol for is what? Well, the only disease we No, not really. That's hydroxychloroquine. Meningitis, we use it in the developing world, but I mean in the US. In the US, for which disease would you use chloramphenicol? Yep, excellent. Yeah, it exists in the form of an ointment, and you can use it topically. It's okay. It's really functional. It's topical, no problemo. You know, that is the one. So, but you don't use it systemically. Don't do that, guys. So, an adverse effect of this drug include anemia, which is dose dependent, and aplastic anemia, which is dose independent. What does dose dependent, dose independent mean? That means that the anemia occurs when you use plenty of the drug, but the aplastic anemia occurs even at low dosages if you have high susceptibility. Can somebody tell me what is the difference between aplastic anemia and aplastic crisis? Yep. So in case of the aplastic crisis, it's only RBCs. In case of aplastic anemia, it's all of them. All cells will be down. It can lead to gray baby syndrome. Basically, the babies, I will show you a picture of a baby with a gray baby syndrome. I hopefully will find it. But it will not be gray baby syndrome. By the way, the babies are not as gray as you think. Okay, they look like this because they have a defect in their UDP glucuroneal transferase. Basically, it's not developed yet in a good way. Okay, so they will not be able to metabolize the drug good. So actually, the drug will become quite toxic for them. Because the drug is super toxic to these people, they can start to develop actual toxicity that can kill the baby himself, because the drug is not getting conjugated in their liver because they have a deficient UDP glucuroneal transferase. So these accumulations in the baby mouth and all over his body, these are basically the drug himself, okay? So be really careful when using it in the children because they don't have an active UDP glucuroneal transferase yet. So the mechanism of resistance is a plasmid encoded acetyltransferase that inactivate the drug. So the acetyltransferase will stop the drug from working. The next drug is a clindamycin, and the clindamycin, oh yeah. So two drugs for acne. We spoke about one drug for acne today. What was it? Doxy. The next drug is, tetracycline, doxycycline especially. The next one is clindamycin. If any of you had acne, I hope you did not take clindamycin. Well, actually, it is according to the protocol. You should, but this thing sucks. Vitamin A is amazing, but it's the last resort. Why? Because it's hepatotoxic, okay? The retinoid, first of all, learn it. Because, I mean, for step 2, you must know this. For step 1, not really. But for real life, you must know this. So, first of all, we use the topical retinoids. Then we use the antibiotics like clindamycin and doxycycline. Then if that does not work, we need to use like the actual vitamin A orally. But before doing it, you have to do two things. If you are a man, you do LFTs, okay, liver function tests. But if you are a lady, you must do the LFTs, but you must do pregnancy test and take 2 types of contraceptives, okay? Not 1, 2 types of contraceptives. Why? Because this drug is highly hepatotoxic and also teratogenic, which means it can lead to serious problem in the baby. In the future, if they ask you why, tell them because it binds to the HOX gene. You're like, what is HOX gene? Well, the HOX gene is a gene that decides that you're, in the future, I will be teaching you in reproductive, you have 46 chromosomes. Who the hell decide that your fingers need to be there? Or your eyes need to be there? Or your foot need to be there? Who decide that? That's the HOX gene. We'll speak about it when we speak about reproductive. And that guy can be inhibited with all-trans retinoic acid, okay? Retinoic acid, basically. So the clindamycin blocks the peptidyl transferase, peptidyl transferase, which is the translocation. That was different one. Remember the clean marsidase. So here it is. Yeah, the long clean, transporting clean marsidase. Yes, transporting clean marsidase. So it blocks the peptidyl transfer, which is in the translocation at the 50S subunit. It's used for anaerobic infection, and there is this rule for surgeon. It does not work for internal medicine guys. So they say, oh, infection above the diaphragm, it's treated with, come on, does anybody know? Above the diaphragm, clindamycin. Under the diaphragm will be metronidazole. Yes, clindamycin. Above the diaphragm. Excellent. Anaerobic infection like bacteroides, Clostridium perfringens, in aspiration pneumonia. Who's at higher risk of aspiration pneumonia? Which population? Alcoholic. Yep. And paralyzed people. Lung abscess. Yes. People in coma. Excellent. Lung abscess or oral infection. All of these can be treated with a clinda. And also effective against invasive group A streptococcal infection, but it's when it's all over the body. Treat anaerobic infection above the diaphragm with the clindamycin. While if it was under the diaphragm, you will use metronidazole. Be really careful. I told you before that ampicillin, amoxicillin, all of these guys can lead to pseudomembranous colitis because of the killing of the normal flora of the intestine and the overgrowth of the Clostridium difficile. Today, I'm going to tell you that is true. In addition, know about the clindamycin. For real life, amoxicillin is the most common agent that causes it because it's the most common antibiotic that is used in the world. But if we speak about the sake of the USMLE, clindamycin is the most commonly asked drug. Okay, they will give you a question about the clindamycin and the patient with abdominal pain, fever, and he has like this pseudomembranes on his colon. You must think about pseudomembranous colitis. Does anybody remember how pseudomembranous colitis is treated? Nice. Vancomycin, oral vancomycin, or metronidazole. Yes. Okay, good. So the group of drugs we are going to speak about include linezolid, macrolides, and polymyxins. So let's do these before the break. Linezolid, the mechanism, it inhibits the protein synthesis by binding to the 50S subunit and preventing the formation of the initiation complex. If you take a look at the initiation complex, we had two antibiotic working, the aminoglycosides and the linezolid. So if that initiation does not occur, you cannot make a ribosomes. No ribosomes, no translation. No translation, no proteins. No proteins, cell dies. Okay. Clinical use for the gram-positive species, including MRSA and vancomycin-resistant enterococcus. Oh, that bit. Well, basically, this one is bad. Super bad. When we see a patient with that, we close him up in the room, and yeah, super serious. Guys, there is something like this, resistance shard, shard antibiotic. So sometimes as a doctor you will get a paper in which has like different names of antibiotics, all of the names of the antibiotics, and it just like comes from the lab, you know, and they tell you, oh well, he's resistant to that one, or is not resistant to that one, stuff like that. If you get the lab back and they tell you this patient is resistant for everything, what you will do with this patient? I mean, normally we are resistant for 1-2 drugs, but let's say that he's literally resistant to everything. There is no antibiotic he's not resistant to. Live in a bag. Okay, that's possible to do, but since we are human beings, we try to help him. You can use, one drug does not work, so what you will do, if one drug does not work, well, if you use each of them alone, they don't work. So what you will try to do is to combine the drugs together. I mean, give aminoglycoside together with a beta-lactam antibiotic. Beta-lactam antibiotic will destroy the cell walls. The aminoglycoside will enter the cell and destroy it from the inside, okay? Try, of course, put it in a bag. I'm not kidding. This is actually really good advice. Don't get near him. The last thing we want is such infection to spread. But in the same time, we need to help him as well, yeah? So we try combination therapy on him. Okay, so we see here something saying something like this. Adverse effect, bone marrow suppression, especially thrombocytopenia, and peripheral neuropathy and serotonin syndrome. Guys, I cannot tell you how much this is important. Serotonin syndrome. Yep. Oh my God. Okay, so what is serotonin syndrome? Serotonin syndrome, this patient have myoclonus in it. So what is myoclonus? They basically start having this stiff hand movement that is stuck at one position and a jumpy type of thing, yeah? So they are full with happiness, they are raising their hands up and right and left and forward. I know this is horrible, but it reminds me of this guys, the secret police in the Nazis. So the Nazis had this secret police that always raised their hand, like Heil Hitler and stuff like that. Anyway, why I'm saying that? So these guys remind me of one thing. They had all serotonin syndrome. They were manic, and basically one more thing. They had altered mental status. They had this rigidity of their hand. And they also have one more thing, which is total disorientation. Okay? That helped me to remember all of the symptoms. So they have hyperthermia. They have delirium, basically, and confusion. And they have myoclonus. Yes, which is the jerky hand movement, the Hitler type of thing. Yep. So that's it, the one. Take it. Okay, so the next one goes something like this. How can the patient becomes resistant to linezolid? Well, basically, the drugs work on the ribosomes. So you use, so the ribosomes themselves need to be altered so he cannot work at them. So by a point mutation, for example. Okay, so the next drug is a macrolide. Guys, I want you to know for the future, if you have a patient who has sensitivity to penicillin, I mean he's allergic to penicillin, and they ask you which drug to give, write macrolide, okay? If the patient is allergic to penicillin, give macrolide. Azithromycin, clarithromycin, erythromycin. Remember, every child who's born gets 4 things and an ointment in his eye. What were the 4 things? What 4 things do we give for a baby? Hepatitis B vaccine, vitamin K, vitamin D, yes, and vitamin K, and also iron. Absolutely, good. Vitamin B12, not mandatory, but the other ones are mandatory. So, okay. By the way, guys, is it good to drink milk if I have anemia? I mean, if I have iron deficiency anemia, should I drink milk? You should not. Bad idea. Let me tell you why it's a bad idea. Iron is a cation, divalent cation. What does divalent cation mean? It means it's a cation that has a +2, yes? And iron is what? It's also +2. So if you drink a lot of milk, which has plenty of calcium, can iron get absorbed? No. So you tell this to your patient with iron deficiency anemia, drink less milk. Don't drink milk. They don't listen, it's their problem. You have just to give the advices. Okay. Nice. Actually, this is a thing. People who think that milk is amazing drink so much milk, they end up with milk alkaline syndrome and iron deficiency anemia. So, the next one. Mechanism: inhibit protein synthesis by blocking the translocation. Clean Mercedes transportation. So clean Mercedes macrolide transportation, translocation. And these drugs, bind to the 23 rRNA in the 50S subunit, and they are also bacteriostatic. Notice, guys, so far all of the protein synthesis inhibitors are bacteriostatic except one. What was the only protein synthesis inhibitor that was bactericidal? Bactericidal. Yes, nice. Okay. I did not expect someone to know that. No. Chloramphenicol is also static. The only one that is cidal was aminoglycosides. That is the only one. Okay. So, if we go here, you can see that clinical use, atypical pneumonia like mycoplasma, chlamydia, and legionella. All of these are intracellular super small organism. Also for STIs such as chlamydia, intracellular super small organism. Gram-positive cocci, streptococcal infection in patient allergic to penicillin. High yield. They give you a patient allergic to penicillin. They ask you which drug to give him. Done. Azithromycin. Okay. And Bordetella pitrusis. What is that? Whooping cough. Absolutely. Good. So the next one is wait. Oh, for a second I thought I stopped recording. That was crazy. Scary. Adverse effect. Okay, macrolide. So the macrolide can lead to gastrointestinal motility issue. Why? Because the macrolide can bind to a receptor called motilin receptor. You're like, but it's not written in the text, man. Are you kidding me? Well, guys, I don't kid anybody. It's here in the GI chapter. We are going to learn together that there is something called the M receptor. That M receptor is responsible for the movement of our intestine. Where it is? Yes, motilin, M receptor. So we even use, see, erythromycin. What is erythromycin? A macrolide. A motilin receptor agonist such as erythromycin can be used to stimulate intestinal peristalsis. Guys, people with diabetes mellitus can have neuropathy and destruction of the nerves that lead to the movement of the intestine. If the nerves are destroyed, you cannot give drugs that work on the nerve because they are destroyed. You have to give something that works from the inside, like erythromycin. You're like, wait. Are you telling me that we use an antibiotic to stimulate the GI movement? Yes, that's what I'm telling you. We use an antibiotic, erythromycin, to stimulate the motilin receptor to lead to GI movement. Gastrointestinal motility issues, arrhythmias leading to prolonged QT interval. Oh yeah, people who did cardiology with me. Do you remember what a long QT interval can lead to? Torsades de bois. It will start to come together now. Each system you finish, the more it comes together. Acute cholestatic hepatitis. Yes. So people about the QT interval or about the motility? Okay. So there is two antibiotics you must know that leads to QT interval. The first one is macrolide, and the second one is the fluoroquinolones. Basically, if we have B wave, QRS complex, and the T wave, we have something called the QT interval, which starts from here until here, okay? So this QT interval has a length which is around 400 milliseconds. Nobody cares about that for now. But let's imagine that we have an ectopic foci, which is a foci that constantly making electricity firing inside of our heart. And it's firing and it's hitting the T wave. The T wave is a refractory period. Since it's a refractory period, can we have any contraction? Yes or no? We cannot. Let's say you used macrolide. Macrolide blocks some potassium channels, which can lead to prolongation of the PT interval. Now the exact same ectopic foci is firing, but now it's firing. Is it firing at a flat line or at a wave? No, not a wave, flat line, flat line. Wave is here, wave is there. It used to fire at a wave, now no wave, yeah? So no wave. If it fires at no wave, it can start one more action potential, which can precipitate to something called torsade du bois, which is dancing around the points, yeah? Which is some type of arrhythmia, basically, okay? So, basically, the azithromycin, well, the macrolides and the fluoroquinolones can lead to QT prolongation, thus the ectopic foci inside of our heart that are normally latent and do nothing can be activated. This macrolide can also lead to rash, eosinophilia as well. Increased serum concentration of theophylline and oral anticoagulant might be needed. We will learn about how this drug will be affecting our cytochrome B450 when we study it. So basically, clarithromycin and erythromycin inhibit the cytochrome B450. I don't want to go in details about this, but basically when you inhibit this enzyme, which is located in the liver, the substances that are metabolized by this enzyme will increase in the serum, like theophylline and oral anticoagulant, which can lead to bleeding disorder. Mechanism of the resistance is methylation of the 23S-rRNA. We cannot consume alcohol. Not this drug, actually. No, not this drug for the alcohol. The one that was for the alcohol, if I'm not mistaken, it was the aminoglycoside, because of the aminoglycoside, and there was one more important than aminoglycoside even. Oh, Vanco. That was Vanco, for sure. So vancomycin, if you consume it with aminoglycoside and metronidazole as well, they can lead to disulfiram-like reaction, which leads to like a hangover symptoms, you know. So you should not do that. If you speak about alcohol, now I'm getting what your thought is. If you are telling me, well, if I take alcohol with this macrolide, will the alcohol become more toxic? The answer is yes, because you are inhibiting the cytochrome B450, which metabolizes the alcohol. Okay, the next drug is the polymyxin, super new. Colistin and polymyxin. So the polymyxin E and B. Guys, if you see somebody with a resistance to this guy, start praying on his soul. Caution, so they're right. Cations polypeptide that binds to the phospholipid of the cells that are gram negative, okay? So they destroy the membrane and they lead to leakage of the cell component, which lead to cell death. It's actually quite basic. It just bind to the wall, destroy it. It's not complicated. It just bind to the phospholipid bilayer and destroy it. You're like, wow, that seems like a perfect, easy drug to use. Yes, we don't like to use it at all because of its horrible side effects. But if we speak about it, we don't use it orally, but we like to use it topically. Because if we use it topically, it will not lead to its horrible side effects. Polymyxin, I like somebody once told me polymyxin has poly side effects, which means many side effects. Clinical use, salvage therapy for multi-drug resistant gram-negative bacteria like Pseudomonas, E. coli, and Klebsiella pneumonia. Polymyxin is a component of a triple ointment that is used to treat superficial skin infection. I hope I will be able to find this one. No. There is one ointment, guys, that is used for that is used for sometimes like impetigo, stuff like that, and it has like 3-in-1 antibiotic type of thing, which covers both the gram-negative, positive, and also cover the it can cover also the anaerobes. which can basically work in everything. I believe it was... yeah. So I cannot find it right now, but basically we use it in a cream, okay? And that cream normally has like 3-in-1 drugs, not only colistin, but 2 other drugs which can make it work against basically gram-positive, negative, and also against the everything basically. Which makes it like perfect drug, and we can use it topically, which means no problem. Oh, here it is. Yeah, so it has tetracycline, colistin, and basically trimethoprim. And yeah, it's used only for topical stuff. Keep in mind that this drug has some really bad side effects, thus we don't like to use it orally, like neuro, and neurotoxicity. The neurotoxicity will lead to slurred speech, weakness and paresthesia, and even respiratory failure. Okay, guys, so we'll have a little break right now for 10 minutes. I will come back, finish up the sulfonamides, dapsone, trimethoprim for you. Hopefully speak a little bit about the fluoroquinolones and daptomycin and metronidazole, as much as we can, guys. So see you guys in 10 minutes. So now we are going to speak about the sulfonamides. Let me just check. Yes, it's recording. So, guys, to make DNA, we have to have one vitamin. Which vitamin helps us to make DNA? Yes, B9. So, if we have any alteration of the process of making us an active B9, we will have less making of DNA. We have less making of DNA, we will have less making of basically active type of no DNA, no cells, no transcription, no translation, nothing. The cell dies. And that's exactly how this drug works. They work by inhibiting the B9 of the bacterial cells, okay? So let's start with the story. So there is a particle called BABA, like Papa, and dipteridine. When they bind to each other, they make the dihydropteroic acid, which is done by the dihydropterate synthetase. And then it becomes dihydrofolic acid, which then works as the dihydrofolate reductase to make tetrahydrofolic acids, which is the B9 that we need to use to make thymidine and DNA. Yes? So if we say somebody does not have, if somebody blocks any of these stages, or of course we are not speaking about somebody, this is a bacterial cells. No bacterial cells will not have thymidine, it will not have DNA. If it does not have DNA, it will die, okay? So let's start with the first group of a drug, which are the sulfonamides. They include the SMX, which is the sulfamethoxazole, and cefixazole, and sulfadiazine. The mechanism of them, they inhibit the dihydropterate synthase. Thus, they will inhibit the folate synthesis, okay? So we have bacteriostatic drug, bactericidal, when you combine them with trimethoprim. So here is the trick. Trimethoprim alone is bacteriostatic. Sulfonamides alone are bacteriostatic. But when we add them together, they become bactericidal. How do we call that? Static makes it cidal. Static with a static makes it cidal. Synergism, absolutely. Good. So they're right. Clinical use. It can be used against the gram-negative and positive. for nocardia as well, and trimethoprim sulfamethoxazole, especially for simple uncomplicated UTI, lower urinary tract UTI, for example, acute cystitis. In acute cystitis, do we have fever? No, we don't. Remember, in acute cystitis, the most important symptoms was what? It was not systemic, absolutely, but it had some symptoms, which were the urgency and the frequency. Absolutely, yep. And sometimes burning micturition. Yes. TNB. So keep in mind that we give this drug just like this is for completeness sake. This is not something you are required to know for your USMLE, but I just like, yeah, this one. Okay. So when you prescribe it to somebody, prescribe 400 by 80. What is that? You are giving 400 sulfamethoxazole and 80 trimethoprim, okay? They come in the same tablets, so you don't have to give 2 tablets. It's basically just like 1 tablet thing. You make this patient take 1 tablet every 12 hours for 3 days, okay? And the UTI will go away. Okay, next, we have an adverse effect of this medication. Wow, we have a list. So this list means that there is a lot of side effects, but to be fair with you, they rarely occur. So hypersensitivity reaction, hemolysis in G6PD because it's a sulfa drug, nephrotoxicity leading to tubulointerstitial nephritis. Does anybody remember which cell is seen in the urine in tubulointerstitial nephritis? Yes, it's possible to see a cast, but the cast will be made out of what? Eosinophils, absolutely, yes. Photosensitivity of the patient as well, Stevens-Johnson syndrome, kernicterus in infant, absolutely. And it can displace other drug from the albumin. For example, it can displace warfarin, which is a bad thing. That means that it will make the warfarin more active, leading to bleeding. So, mechanism of resistance, they alter the enzyme himself, the dihydropterate synthetase. Or they decrease the uptake or increase the BABA synthesis. Basically, yeah, you are blocking this, but if you keep increasing the substrate, it will use it in somehow to go downstream. So more substrate, it will use whatever left of the enzyme to keep going. So in case of dapsone, it's another drug. It's also a sulfa drug. It looks like sulfonamide, but if we speak about the name, it's also like sulfonamides. I mean, if we speak about the fact of what we use it for and all of that, but in reality, it looks a little bit different structurally. We use it for leprosy, for both liberamatose and tuberculoid form, but to be fair, it's more for the tuberculoid form because the liberamatose form had another drug that its name you must know. It starts with, yes, clof. I actually cannot, clofazimide or something like that. I cannot know the name. I just remember that it starts with a clof. So, pneumocystis jirovecii prophylaxis, as we said, when the CD count goes under how much? $200, and the 100 was for what, guys? The 100 was for what? Toxo, absolutely, good. So a retreatment when used in combination with the trimethoprim. So the adverse effect include hemolysis in G6PD deficient patient. Also, it can lead to methemoglobinuria. Keep in mind, nitrates can lead to that as well. It's a whole topic we are going to speak about a lot in respiratory, the methemoglobinuria. And the granulocytosis. Okay, so this dapsone drug also work at the same step as sulfonamide did, and yep. So the next drug we are going to speak about will be the trimethoprim and the pyrimethamine, which work at the dihydrofolate reductase. Both of these are bacteriostatic, but if you give them together with sulfonamide, they becomes bactericidal, which means it's a synergistic effect. The clinical use. It's used in the combination with sulfonamides like trimethoprim sulfamethoxazole as the anti-UTI agent I just showed you. Remember, 3 days, 1 tablet every 12 hours. Causing sequential block of folate synthesis. Combination used for UTIs, Shigella, Salmonella, BCB, BCB, pneumonia of course, and prophylaxis for Toxoplasma prophylaxis as well. So for the BCB prophylaxis, when the CD count goes under 200, and for the toxo, when the CD count goes under 100. Yeah? So an adverse effect include hyperkalemia at high dosages. And well, I have a question, guys. Why do you think it leads to hyperkalemia? Just take a guess. If you want a hint, it kills the bacteria really quick with high dosages. Well, basically when you destroy the cells, some of the potassium inside of them will come out, but it needs to be in combination to be like a killer. So megaloblastic anemia can be seen because remember, this drug after all is leading to which vitamin deficiency? B9. It can lead to leukopenia, granulocytopenia as well. Basically, all of them bnia, and the main cause is the decrease in the vitamin B9. Okay, so which may be avoided with co-administration of a leucovorin, which is folinic acid, which will, you're like, okay, wait a second, this is confusing, man. You just told us that the main idea here was to reduce the vitamin B9 and to get less DNA, and that's how we kill the bacteria. And now you are telling me we give the patient folinic acid not to get B9 deficiency. I mean, are you serious, man? It's not making sense. Well, remember, this process, is it in the bacteria or in the human being? It's in the bacteria. So my task is to reduce the B9 in the bacteria and to give a form of vitamin B9 to the human being that cannot get into the bacteria. So that we call folinic acid, okay? Thus, the patient kills his bacteria, but he does not kill his own B9, okay? So the next drug is going to be fluoroquinolones. So fluoroquinolones, all of these that ends with cine, ciprofloxacin, enoxacin, norofloxacin, olefloxacin. Wow. And the respiratory fluoroquinolones like chemofloxacin, levofloxacin, moxifloxacin, floxacin, a lot of floxacin. They inhibit the prokaryotes enzymes topoisomerase 2. It's also called DNA gyrase and topoisomerase 4. Does anybody remember what was the function of topoisomerase? Absolutely, it helped to remove the supercoils. So when it removes, the topoisomerase removes the supercoil. So when we inhibit the topoisomerase, it will not be able to remove the supercoils. So there will be entanglement, which will lead to less DNA replication and thus less DNA expression. Yeah? Gene expression, less gene expression, which lead to the death of the cell. You should not take it with antacids because it makes it, well, basically useless. Clinical use, a gram-negative road of urinary and GI tract, including Pseudomonas, some gram-positive organisms, and otitis externa. Okay, so let me give you, yeah. So guys, from time to time, I will just be giving you some general stuff that you must know. It's not because you are doctors, because you are human beings. You see this every day, you know? Okay, so if someone has like otitis, you can use this ciprofloxacin for the eye, and the exact one you use for the eye, you can use it for the ear. The same one, the exact same one. You tell the patient to put 2 drops, 3 drops once every 12 hours for a week. You're like, wait a second, why for a week? Well, to be real with you guys, it take him 1 day for the symptoms to disappear, but why you are giving it for a week? So during this period, it will not reoccur. It will not reoccur. Okay? Okay. So they write, gram-negative rods in the urinary and GI tract, including Pseudomonas, and we use it against the Pseudomonas, that's the otitis basically. Some gram-positive organism, and also for otitis externa. Cystic fibrosis patient love fluoroquinolones. Adverse effect, GI upset, superinfection, skin rashes, headache, dizziness, a lot of stuff. And less commonly can cause, this is the important stuff, leggy cramps and myalgia because of the electrolyte disturbances, okay? Contraindicated during pregnancy or breastfeeding in children less than 18 years old. Okay, and in children less than 18 years old, because it leads to damage into the cartilage. Which other medication we spoke about today, I mean an antibiotic that also was leading to a problem in children and the pregnant lady, doxycycline. But doxycycline or tetracycline was leading to bone problem. Fluoroquinolones lead to cartilage problem, okay? Some of them may prolong the QT interval, and if we prolong the QT interval, we might have what? Torsades de Bois, absolutely. Mechanism of resistant chromosome encoded mutation in the DNA gyrase, okay? Also, plasmid mediated resistant, again, efflux pumps. We have a creation of efflux pumps, and these efflux pumps will bump out the drug outside of the body. It may cause tendonitis, which is inflammation of the tendon, or even tendon rupture, especially in really old people, more than 60, and in patient taking prednisone, and especially like people with lupus or something like that. Ciprofloxacin inhibits the cytochrome B450. Remember what other drug led to that as well? Inhibition of the cytochrome B450. Macrolides, absolutely. Nice. Okay, so daptomycin mechanism, it's a leopeptide that disrupts the cell membrane of the gram-positive cocci by creating a transmembrane channel. I want to show you a picture of that. Basically, it creates a channel in the cell, in the bacterial cell wall, which lead to osmotic damage, basically. So that osmotic damage lead to, well, a lot of fluid moving through the cell membrane, which lead basically to the death of the cell. So all of the potassium will go outside of the cell, and all of the sodium will go inside of the cell, which lead basically to the damage of the bacterial cell and its death. So let's take a look. Yep. See, daptomycin is complexing with the calcium, then it comes to the cell wall, disrupt it, make a hole, pass all of the potassium to the outside, lead to osmotic damage of the bacterial cell. That's daptomycin. So the daptomycin, it's used for Staph aureus infection, especially MRSA, bacteremia, endocarditis, and the vancomycin-resistant enterococcus. Not used for pneumonia, vividly binds to, oh yeah, because it's inactivated by the surfactant. Yeah, daptomycin does not work in the lung, basically, okay? Because it binds to the surfactant, super important. It's one of the rare antibiotics that lead to such symptoms such as rhabdomyolysis and myopathy. But do you remember which was the other antibiotic that also led to neuromuscular blockade? Yep, it was aminoglycosides, absolutely. That led to neuromuscular blockade, and it was contraindicated in case of myasthenia gravis. The last drug for today is metronidazole, and the mechanism of it is the creation of a free radicals metabolite in the bacterial cell, which damage the DNA of the bacteria and kill it. So it's not only the bacteria, the metronidazole is actually not an antibacterial, it's not an antibiotic, it's an antiprotozoal. It was created for a protozoa, but it kind of acts on some anaerobic bacteria, thus we use it for it. So it used to treat Giardia, Protozoa, Entamoeba, Protozoa trichomonas, Protozoa gardnerella bacteria. Anaerobics as well, like Bacteroides and C. diff. It can be used in a place of amoxicillin in H. pylori if the patient was penicillin allergic, especially in the triple therapy. What is a triple therapy? It's when you give 3 medications to treat the H. pylori, which are amoxicillin, macrolide like azithromycin, and the BBI. Absolutely. Good. So they write, it treats anaerobic infection below the diaphragm with metronidazole, above the diaphragm with the clindamycin. The side effect is disulfiram-like reaction, which lead to severe flushing, tachycardia, hypotension with alcohol. Basically the hangover symptoms. Also a metallic taste. Does anybody remember which other antibiotic led to disulfiram-like reaction? Aminoglycosides. Done. Okay, good. So that's it for today. We did 7 pages. This is really good. Tomorrow we'll come back. We will doing the same thing. I will finish the last 2 pages from molecular, and then I will come here, push this page, this page, this page, this page, and hopefully this page. So see you guys tomorrow, and we will be back at the same time tomorrow. Goodbye, everybody.